Okay. Welcome back, everyone. Hopefully you've been having a very good hackathon. I know I have been having a very good hackathon. It's been a lot of fun and I've learned a lot. So now we're going to conclude by going around the room, around the virtual room. Lydia is going to help us do that. And folks are going to share what you did briefly, if you'd like, what you learned, what you liked about the hackathon, and possibly what you would like to do differently next time with the hackathon. So I know we had a few questions. We're spending time based on what kind of time we had left and when people will be around. We have about 16 people on the call, so we're planning to go up to 2 hours. Hope to end before the next 2 hours, but that gives everyone 5 to 10 minutes of time to discuss what you'd like to do on those topics. I'll let Lydia introduce folks. So Lydia, I know, for example, some folks have to leave early, like the ants folks, and so maybe we can start there.
Okay, let's start the ants folks, and then I know some of you are paired up, so you can talk together if you like. First ants folk is Min. Hello.
Sounds good. Share the screen. Okay, great. Yep, so I am Min. I am part of the Pixel group and the ants folks, as Lydia refers to us. And I'll be talking a little bit about the framework we've been putting together for the FMOS to CCF alignment. So I think, so we actually had a little bit of an advantage in that we've already been developing an FMOS to CCF approach using an ants-based framework. And so the goals for this particular hackathon for us was actually to adapt this framework into kind of a containerized pipeline that can be very portable and easy to run on Bill or other kind of similar systems. So to kind of give a quick overview of the framework, the main idea is that what we were finding is that due to the kind of large morphological differences between FMOS and CCF, we're finding that kind of direct registrations just really wasn't performing very well, at least without some assistance in terms of landmarks and whatnot. So our approach that we've been developing, the idea is to use an intermediate atlas as kind of like a halfway point to register new FMOS images into the CCF. So this average atlas is constructed from other FMOS images, and I hope is that they kind of capture a lot of the morphological characterizations of FMOS images, such as like the enlarged ventricles and shrinkage of particular structures. And then what we did was we performed an atlas-to-atlas registration between this new FMOS atlas and the CCF, and with the help of some annotations that the Allen team was able to help us provide. And I should note that this particular part of the framework is something we do one time. So we actually put a lot of effort into making sure that the various labels and landmarks are registered very well. And this framework, when it's being run, doesn't actually uses kind of a pre-built alignment, the kind of canonical transform between these two atlases. Then with any new FMOS images, then we can just use actually a standard registration from the FMOS into the average template space and then concatenate the transforms to get our CCF alignment. So this is our FMOS atlas. It's constructed using 30 unique specimens, which we then left-right flip, so about 60 images. And we constructed this at 25 micron to match the 25 micron CCF atlas. This was done using 3 iterations of the ANTS template construction tool. And then, these are the various structures. I think there actually is a couple, maybe one more structure, but this shows the structures that we use to perform the alignment. Because we're actually performing an atlas to atlas alignment, we can actually work really hard to make sure that the various structures line up very well. And you can see that between the 2 atlases, there's in general overlap of over 0.9 diets for most of these structures, although some of these smaller ones are still kind of difficult to get together. So here is just a slide showing the alignment between the atlas to CCF, or the FMOS atlas to the CCF. And you can see that a lot of the morphological structures are much better aligned than we've seen previously, particularly kind of the shrinkage of the ventricles to match and various other substructures. So for the purpose of this hackathon, what we wanted to do then is to take this framework and implement it essentially on Bill as a standalone pipeline. I think I mentioned a little bit about how to do this with CWL wrappers and earlier this week, but the main idea is that we can represent our test input as these YAML files. That's what these things look like. Essentially, they're just they're text files that describes an input image, in our case, an atlas directory, and then we didn't have this for this particular hackathon, but a neuron reconstruction directory with some SWC files as the placeholder right now. But then we can take this and essentially send the input through the pre-processing, which then gets sent to the registration, and then the transforms out of the registration is used in this 3rd module that transforms the neuron reconstructions. For the pre-processing, one of the things that we found was pretty key for getting good registration was that we really had to get rid of these stripes prior to the registration. We were finding that these lines were getting matched to actual boundaries in the CCF, and it was causing kind of...
I was going to say that. Sorry.
Yes, go ahead.
I didn't know my mic was on.
Oh, okay.
But I was going to say that in my presentation.
Oh, okay. Well, I'm looking forward to hearing about it. But for us, we took for a domain approach, essentially just performing, looking at it, we can see that there is peaks associated with these stripes in the frequency domain. So we developed these notch filters after detecting these peaks, and it does seem to remove the stripes for the most part, and while preserving a lot of the structures that you would expect to see in the FMOS image. For the registration part, this is pretty straightforward. It actually just runs ANTS. We take the pre-processed image, register it to the FMOS image, FMOS template, and then concatenate the 2 to get our final transform. So here is an example of an individual image this time registered to the CCF. We can see that the alignment doesn't look as clean as the atlas to CCF alignment, since this is an individual subject. But we can still see that a lot of the structures that were kind of deformed relative between the modalities is much better aligned now after performing this registration. Here's another slide showing some similar features. And then here is the CCF label placed on top of the registered FMOS image. And I guess it's a little bit hard to see, but a lot of these structures seem to at least look like they line up with the FMOS, although it's a little bit hard to say. And then finally, this last part, we didn't have any SWC for this particular challenge, but we've had SWCs in the past that we've been able to transform with the registration results. This is just like a quick example, I guess I can go through. So this is what one of these neurons look like going through the original FMOS image. Here we've taken on the right here, the registered image, and apply that transform to the same neurons. And now you can see the neurons overlaid into the CCF or even on top of the CCF label. So you can see what structures of neurons are going through. So yeah, just to summarize, this is kind of the package that we put together for this hackathon. It contains an image file for the Singularity container. So this is the kind of like the main bulk of where everything's being called, and then wrapped around this container is a couple common workflow language wrappers, essentially describing the pre-processing tool, the registration tool, and the transformation tool. And then all of that is put together as a single pipeline for the alignment. And then this is the test image. So if you want to run this, you can run this particular line pretty much anywhere. And it should hopefully give you the registration between the FMOS and CCF. Okay.
Cool. Min, do you want to make sure that that you leave all the in our output directory that there is enough to reproduce what you've just shown here, like your image and those CML and the YAML file? That would be great.
So I should be able to just copy this into the directory, and I think anyone should be able to run it.
Yeah.
From that, and that's kind of like the advantage of the containers. But yes, I'll make sure to, maybe you can help me try it and reproduce it from the package.
Questions for Min?
I have a question.
Sure.
So when you guys did the segmentations, you showed you guys used masks of like 8 or so, fairly large structures. I was wondering how you guys, how you guys decided on those structures, and particularly, is there a reason you're doing those?
I'm going to answer that one. So the first one was then from our first registration between the template and FMOS, what was going on wrong. Second one was then our anatomist gave us a list of things that are visible to him that he's willing to draw with confidence. And so we started at, around the cortical area, let's fix that. And so now we're got into the hippocampus. Let's fix that as well. So this was then, as we were looking at it, what is causing the difference? What do we want him to draw? And sometimes he doesn't draw everything. So I actually, to match his drawing, sometimes I have to slice and dice the CCF or to add and subtract bits to match his drawing. So that was
cool.
That was my task.
Gotcha.
Oh, all right. So should we move on because of time? This is cool, and we've got this recorded. Who needed to leave? Jeff, were you the one that needed to leave, or do you want to go quickly?
I can go quickly. Yeah, I wasn't running any registrations. I was really just here to sort of help with the initial tutorial early on, and help Min figure out the build system. But I did think the hackathon is very useful, and a lot of the ants birds of a feather session was really useful to get a lot of different very knowledgeable people in place at the same time to talk about some ways to work on that package. So I definitely think that was very, very helpful. So yeah, I think overall it was a very good experience.
Cool. And then now I'm going to go across my screen. Daniel, you're the first one across my screen.
Hello, I'm going to share my screen for a second here. Sorry. So I decided to stick to the questions that Matt asked us in the chat in a fairly straightforward manner. So let me just skim through a few things. So what did we do at the hackathon? We shared our standard pipeline for running LBDMM on different kinds of data sets. I know there were a couple people who were interested in using it on light sheet data and other data sets. So if anyone did end up using it, I'd love to hear feedback on if it was possible, if you ran into trouble. We shared some examples of our optimization approaches that don't use gradients. And we implemented a registration of an FMOS challenge data set, which we described in the tutorial. Currently, we're working on wrapping it up into a reproducible pipeline the way that's described on the website. We have a little bit more work to do on that hopefully will be finished today. We learned a little bit about FMOS data and the challenges working with it in particular. So one issue that the ANTS folks discussed also is this idea of using an intermediate template because the structure is so different from the CCF. So we've been working on a slightly different approach for constructing intermediate templates that we believe are a better match to the FMOS data set. And so what we've done here is select a family of structures in the Allen Atlas and allow them to grow or shrink as we define, while allowing minimal amount of distortion in other regions. And by minimal amount of distortion, we use a divergence-free or volume-preserving velocity field here. So this defines a family of atlases, in this example indexed by one parameter, but we're generating families with a few different parameters that we believe are going to correspond to better matches to FMOS data sets. And these transformations can be inverted, but they're not necessarily smooth, which means we would never estimate transformations like these ones in our pipeline. So this is kind of what we are going to emphasize working on going forward, given everything we learned about this type of data this week. We also learned about dataset standards and what we can immediately commit to implementing. So I'm sold on the idea of specifying an affine matrix for every data set. That's 12 numbers as opposed to just 6 numbers. And also naming all our axes, which are 2 sort of separate compartments to this reproducibility in working with data sets. Here's what I really liked about the hackathon. So I'll start by saying, traditionally, I don't like hackathons because I'm very slow at doing things. So it's difficult for me to commit to having to do something in 1 week. But I thought it was nice to have a few milestones for myself and my students to say, well, you know, our repository needs to be public at this time. Therefore, let's finish by this day. It's nice to have those types of opportunities to kind of benchmark yourself. And I really enjoyed discussing important data sets from a lot of different perspectives. So mathematicians, biologists, computer scientists, and having some great conversations with other experts. So I spoke with people from ANTS yesterday, with Lydia and Matt and other people working on ITK and trying to establish standards in registration. So it's been a great social time, as social as it can be, with my mask on, sitting in a small room. Next, I'd really like to do something like this in person. And one thing which didn't get covered, as opposed to, except for one sentence on the website, was what is an appropriate open source software license for this type of work? I'm really not sure. At this point in my career, I'd really like attribution when I produce software. It's not clear to me what the best license is that kind of asks, you know, please feel free to use this or modify it, but please cite my paper. So it'd be nice to have a bit of a discussion about that going forward. Okay, that's all. Thank you.
That's awesome. Should we continue? Ricardo.
Hey guys. Yeah, I think I've had a lot of fun the past couple of days. I've been here mostly for the discussion, but I will be playing with a bunch of the tools you guys introduced over the next couple of days and weeks. Hopefully I was able to provide some representation from the light sheet community, and hopefully you guys found that helpful and it helped frame things. Yeah, so I use registration tools pretty much every day when I analyze my data, but I don't get to interact with you guys and the larger community working on registration techniques. So it's really great to see kind of what everybody is working on and also what great things you guys have coming down the pipe. It was really great to be able to talk to the ANTS folks, because that's what I use every day for my registrations. It's great to hear ways that that I can improve and that the package is improving. And it was also really cool to hear about things that I normally don't think about, like the data standards for how to store how to store transformation data, which I'm 100% on board with with everyone here on. Yeah, I think it's been a great couple of days. I guess, Matt, you asked things that could be better in the future. I think it'd be great to have a bigger in-person presence, but obviously given the state of the world, that isn't too feasible right now. But I hope we can do this again and have everyone in person.
Cool.
Oh, icon registration stuff was super cool too. We'll be playing with that a lot.
Should we do the UNC room? Do you want to go around the room?
Sure. I don't know if, yeah, we have chairs there, but maybe I'll start and we'll go around the half circle here. So I had a wonderful time. I'm so happy that everyone came. I had a lot of fun and I learned a lot. So I went to all the tutorials, which were very beneficial. And I think if you miss some of the tutorials, we have them all on the website, and they're there for you to go back and look at the content. There are Jupyter notebooks for many of them, and there are recordings too. I think we have for most of them. I also went to the BOFs, which were excellent, really excellent. And we also have some notes on the hackathon, in the hackathon repository. Try to keep notes for most of these BOFs if you missed those. I learned a lot about Bill and all these data sets and what everyone else is doing. One project that I wanted to share, I was looking I'm working on creating a web visualization tool for registration of these brain images. And it was working, so I tried it working with the Bill data, download.brainlib.org, and that's working. It's nice to see that going. It was working with these larger nifty data sets, the Allen CCF, and some of the light sheet nifties that Ricardo shared. There is some ZAR support in the viewer, but it needs to be updated to the specific updates that came in the recent standards of the format, and then how it was collected in Ricardo's data. And another kind of lesson learned here is that the FMOS data, I tried with the FMOS data, and it broke the viewer, which is not surprising. But I also learned that, even if it wasn't working for the FMOS large data sets, it would be a poor user experience to download that full data set to decompress it in one go. So I think in the future, maybe a request is to have that as this multi-scale chunked, and GFF would help with that too.
That can be the next hackathon.
That could be the next hackathon.
We can have them in those. We can all learn how to use the chunk format.
Yes, good idea.
Out of curiosity, Matt, did you try using the ZAR files? Because those had the pyramids inside them.
Yes, and I tried, and I think we'll continue working on that.
Okay.
Thank you for putting those out there. Yeah.
Going around the UNC room here. This is Tom, but I just wanted to say I had a good time also attending all the workshops and everything this week. Really great to learn about the tools and the ecosystem surrounding image registration here. I spent most of the hackathon tinkering with the ITK infrastructure around the FFTs that I discussed before, and some of the Gaussian blurring and multi-resolution pyramid generation that we use for our registration pipeline. Was able to use the Bill workshop 2 access and the GPU hardware there to test out and kind of compare these GPU FFT speedups, and was seeing up to 92% speed up of accelerated FFTs on GPU versus the typical CPU implementation that we've been using in ITK. So that's really exciting and really promising for moving forward with some accelerated blurring and just being able to increase the performance of our pipeline there. So had a great time this week.
I think most of what I got done was I was able to present my ideas and push forward on transform interoperability. And that's like 10% me and 90% you guys for listening to me and be willing to sit through it and also push forward like your own ideas and the interaction with the next generation file format. I had a great time. And I'm also hopeful that maybe deep learning registration, it's not there yet. I didn't get it working on the FNOS data, but we'll see in a week or two.
Say your name.
Hastings.
Okay.
I don't think you are in the shot of the camera, so you can see.
That's
Jen, I'm here. I mostly tried to attend all the events, the tutorials, the rules of feather, and so forth. I didn't get a lot of work done. I was excited to hear that Elastics is now or soon going to support ITK transforms, so they can be used without having to resort to transformix, and they can be realized in Slicer and so forth. I need that for an ongoing project, so I'm excited to get back to that once that's ready.
Thanks. This is Paul here, and I appreciate the practical tutorials quite a bit. As an app developer, it's really nice to see that you guys are trying to make your work accessible to lay folk. The new work with OEM and GFF format.
Who just spoke?
There's Paul. Paul
Oh, okay.
and
Oh,
this is Ibrahim in the UNC group. I really learned a lot from the tutorials and presentations. I especially learned a lot from the presentation just now, the ANTS group, about their approach to AppLost. I sort of like, as soon as I got into the world of medical image registration, I dove right into the deep learning end of it. And I didn't really learn all of the huge history and set of other approaches that there are. So this really like expanded my view of what there is.
That's everyone from UNC.
That's all of us here.
Brian.
Hey, everybody. I definitely learned a lot from this hackathon. I haven't been able to participate as much one-on-one. I've had a sick kid at home for the week, but that's just kind of how it goes sometimes. Yesterday, actually, I was catching up on some of the stuff that I missed, and I noticed there was some good discussion about transforms and orientation, and it's something that I've thought a lot about in the type of work that I do. So I wanted to just share really quickly my screen and show just as sort of a, I guess, like a solution that I came up with a while ago for dealing with orientations of things. So can everybody see my screen at this point?
Yes.
Okay.
Cool.
So this is a tool. It's not an open tool. It's just a tool that I build for MDF Bioscience. But we have a cool way of dealing with orientations of volumes that are being loaded in. So Here, I'm going to click on my change orientation button and take a look through. So there was a question that was posed, I think, that Lydia, you had this question about how do you specify for a brain what the orientation of a particular image is, how it's been specified, and there was somebody had proposed, like, maybe you just ask them questions, and actually I already had this set of questions. This is how we do brain orientation. So when you're imaging something, or if it's either a whole brain image or if it's done in sections, typically the person who's doing it can answer the question of which orientation did you start with? What are you looking at when you either scan or when you section your brain? So the first question is, what plane did you cut the brain at? Is it coronal, sagittal, and transverse? In this case, the image that I happen to be loading is coronal. And then the question is, which way did you scan the brain, or which way did you section the brain? So with mouse brains, a lot of times the people that I work with talk about rostral to caudal instead of anterior to posterior, so different medical versus research groups. But in this case, it was a rostral to caudal. And then the question is, what does the brain look like on your screen? Or let's see, we asked the question, what's at the bottom of your image? For this particular image, when it was loaded up, it was the left side of the brain is at the bottom of the screen. So when you go through those three questions, that's enough to know how that brain is oriented, and then we can orient it to the atlas by knowing what the orientation of the atlas is. So we can build up that transform. Thank you, Lydia, for a very challenging data set. This stuff is hard to register, definitely really difficult. And so I kind of have like a half-baked start at it, but it's been really fun. So I'm going to stop sharing my screen at this point. But I definitely learned a lot about a lot of different tools that are out there, and really had a great time. So I hope to be able to join again in the future. Yeah.
Cool. We need that little viewer. That's what we need, but a little simple one that just does that. All it does is that. What did you do? Tell us what you did. Yes. Yeah.
It was fun to work with a couple of scientists on that just to get that orientation, because it's so important. It's so incredibly important to get that right. So yeah.
Cool. Yeah. Okay. Next on screen is Iana. Oh. We can't hear you.
Now?
Yes.
Oh, yeah. Cool.
Yes.
Thanks, everyone, for organizing this hackathon and letting me participate. It was amazing, truly amazing. I'm happy to meet new people and to discover all this variety of packages that are out there. The only thing I wanted to note, as a user of this package, I'm not developing any algorithms myself, but I'm just using your packages. And what I would like to have is more extensive documentation and tutorials for dummies, because an average biologist usually doesn't have a degree in computer science and doesn't know a lot about this registration problem. So it would be helpful for people to have something like these containers that we are trying to produce, just to run it and answer specific scientific questions with this. So now to what I've done, let me share my screen. Can you see the screen?
Yes.
So I put my contribution to this reproducible resource folder, and I think I'm the only one here so far. I'm not sure if it's the correct folder to put everything. So what I did, I used the existing package called Brain Reg, developed by Adam Tyson, who is here. Adam, thank you so much for the package. It's wonderful. It's very, very well documented. So if you see the GitHub page, you see all the explanations of every step that you need to do. And also, this is the best documentation that I've ever seen on the open source project so far. So if you want to do something for everybody to use, just aim at something like Brain Globe is doing. Sorry for unsolicited advice. Okay, so what I did is I adapted the Brain Reg package to work for FMOS data. I added some pre-processing. And I packaged it to work at Bill. So I created a repository which I installed at Bill afterwards in the Singularity container. So just 1 second. This is my Singularity container definition, where I clone and install my repository from GitHub. And this is the very dummy CWL workflow, just with one command, where everything is put together in the command line. And so I also prepared in my repository at GitHub, the link to which is this reproducible resource page. I prepared an example notebook of the pre-processing, which is like the most valuable part here. So what I did is I tried to put the mask around the brain to subtract all the background, which is distorting the registration. Also, I used the two-dimensional FFT on each coronal slice to remove the stripes. I didn't have a lot of time to do that, so I still have some artifacts left. But the stripes are mostly removed, and the brain reg performance actually improved a lot after this pre-processing. And I've noticed that for our confocal clear tissue images, the pre-processing is just crucial for any registration algorithm, because our data is quite different from the atlas. It's sometimes very noisy and low contrast, and has also these stripes from ribbon scanning method. And then I just launch the brain reg through the command line. That's it. Thank you.
That is super cool that you're able to go with these documentation that lots of people had to do very quickly for you, enable for you to like run it on a resource and put a container together.
Well, this is what I could do in 3 days. That's why the pre-processing is so bad, but hopefully I will improve it soon.
But now you have a framework, so now it's about just modifying that part. This is very exciting. That's the dream, that people who wasn't the builder of the algorithm can come and plug and play all these different tools together.
Yeah, that's the goal.
That's awesome. Who's next? Alan.
Hi, everybody. Actually, this is good timing. Yana, I just want to, shout out to her as a huge thank you. So we actually brought Yana into the lab just in November, and she's fantastic and has made a ton of progress. And I think this was a huge contribution, and thank you so much. I should also give a shout out to Adam Tyson as well. We've never had an opportunity to meet, but thank you so much for the really wonderful software, and we've been making a great use of it. In general, I haven't had an opportunity to participate as much as I'd like. I've mostly sat in the background and listened to the really fantastic presentations. And thank you so much for the wonderful group who's come together and taking the time to really explain various packages, registration methods, and even a discussion of various methods of imaging modalities and how they might be used to align to the atlas and the various problems. I think that's been really helpful. Thanks so much, everybody.
Cool. Next up, Lee.
Hi, Lee is my partner, and we are a team, and I can share my screen and share our team's work to you. Actually, it is our previous work, but through this hackathon, we met many different data sets, like the FMOST and the last sheet data set. So it is an opportunity for us to solidify our pipeline. And in this hackathon, we changed our we modified our code a lot, and the pipeline will be changed a lot for adapting the new data, and we developed the GPU version of this pipeline. Also, we generate some meaningful results for have most data set and the last sheet data set. So I want to go through our pipeline and let's start now. Our tools names Brain Aligner, and our advisors are Professor Peng, Professor Qu, and here are some of my team members, and I'm the presenter, Zhixi. Our pipeline consists of 4 modules. They are data pre-processing, global registration, brain region segmentation, and the local registration. And because the image quality will affect the accuracy of subsequent registration, it's necessary to pre-process the images to make sure the texture is clear and reducing the noise. So the pre-processing is divided into 3 parts: artifact reducing, brightness enhancement, and the strap removal. We noticed that there are a little bit artifact in the background, so it can be simply removed by thresholding, and some of the images have the low brightness. Some of the images have the low brightness, and we will take the grayscale value between the 1st percentile and the 99th percentile and rescale them to 0 to 255. Here are some examples. We can see that the dark images are bright enough to identify the texture, and the same operation doesn't affect other images of normal brightness. According to the periodic characteristics of the strap noise, we observe the Fourier spectrum of the image, find the noise which is symmetric about the arranging, and then we construct a Gaussian notch filter to remove the strap noise. It's quite similar at the means presentation, but there are some difference about the Gaussian notch filter, and this is the gap between our work, because I think our data is not smooth at means results, but it contributes a lot for next registration. By now we have finished the pre-processing part. Then comes to the global registration module. We designed a robust point matching based based automatic global registration. In the existing global registration algorithm, they require manual assistance or are not robust and efficient in handling mouse brains with excessively different directions. In Brain Aligner, global registration is automatic. Here the workflow. If we want to align two brains, we need to generate the point site that can represent the brain shape. Here we applied contour detection and generate the brain outer contour. We normalize the coordinate of these 2 point sites, and then PCA-based rigid transformation can solve the huge differences between the brains, even of the brains had the most close to 180 degrees difference in their orientations. Afterwards, the affine transformation matrix is obtained through RPM algorithm, so that the mouse brain has the same size and the direction as template. The affine transformation matrix contains these 4 transformation parameters: scale, translation, rotation, and shearing. This video is the whole process of global registration. Before the local registration part, we conduct the segmentation. About 8 major brain regions by 3D Unite model. We feed brain images and semi-automatic registration results at the training input and target label, respectively. Note, we didn't use the computation dataset as training data. The segmentation results will contribute to the optimization of local registration in next step. Actually, it is an objective metric, and it can get objective function to converge to a more optimal field. The local registration always plays a decisive role in the overall performance of registration pipeline. The local registration can be described as an energy minimization problem, and it can be divided into 2 parts for optimization. We defined voxel classification, but we defined voxel classification probability in subject image, and there is no need for subject landmarks. Because it will find the optimal position that is related to the corresponding target landmark. As to the deformation function, we choose the smooth thin plate spline to model the spatial transform. These 2 parts will be optimized alternately, and all of the optimization will be constrained by 3 features: the gradient, histogram of oriented gradients, and segmentation probability of brain regions. We can observe the movement direction of the landmarks more clearly from this video. All of the compiled files are packed up in the singularity image, and the pipeline is constructed by CWL2. And we can run the configuration files in command line, and it works. But right now I haven't integrated all of the CWL files together, and I will maybe in next 2 days, I will finish this and upload the pipeline to the build server. And here are some results of 2 data sets. Let's take a look. And there are some discussions about the pros and cons. This is the data set 2 last sheet data set. You can see this is a part of missing tissue, but it's still the algorithm it still works. Okay, let's take a look up for this image. It's not a standard half brain image, but the result is still good. And there is no obvious round deformation of the contour. And this example shows the inner texture, the texture of the inner brain can be well aligned. So that's how our team work. And thanks for listening. Also, I want to share what I'm learned, and actually it is very useful for me about the tutorials, because I started to use the CWL tools and the singularity image in our lab. In our lab, it's quite a problem about the servers develop environment, because different softwares can be conflict each other, and if we use the singularity image or the Docker image, we can solve this problem easily. Yeah, that's all.
Good job on the light sheet data. I look forward to checking out the output. It looks like you did a good job addressing some of the challenges I left in for you guys.
Thank you.
And I'm glad, even though you guys are already really familiar with the FMOS data, that you got, there's the pipelining and the CWL part that you can still come and learn and share. So that's really good.
Yeah, we'll complete the CWL part and as soon as possible. Yeah.
I have to leave for like 10-15 minutes. I'm going to hand over hosting duty to Jean. All right. Well, you are host. All right. I'll be back soon.
stop sharing. Oh.
I think Adam is going to go next.
Yeah, I don't really have much to share because I unfortunately haven't been able to come to much of the hackathon. I've kind of been just dropping in and out. But it's really nice to see people kind of working together and collaborating, because whenever you try and publish papers on this, all the reviewers always just ask you to show how rubbish every other tool is and how yours is so much better. So it's nice to see that people everyone is interested in working together and kind of developing the best tools and releasing them so people can use them. And it links to what I've been thinking, what we've been hoping to develop is kind of tools that allow people to just essentially pick and choose what the best algorithm for their data is, and also hopefully kind of prevent algorithm developers from having to write lots of boilerplate. So kind of the more that we can work towards common open standards, both for data, but also for kind of representing transforms and things like that, the better. Yeah, and I hope these hackathons continue. And yeah, because I think it's a great idea, and it's been great to, for the few chats I have had with people, have been very useful. And it's also great to see people using some of our tools that we've been developing. So thank you very much.
Okay. Bryson, do you want to go next?
Sure. For those who don't know, I'm Daniel's student. I think he covered what we did pretty well. But I really enjoyed learning about the variety of different approaches and algorithms to the same kind of challenging problem. I've been working on our pipeline, so I was really interested in the containerizing and CWL, and just learning how people share their code and make it reproducible. So that's what I enjoyed.
Thank you. Greg, do you want to go next?
Okay. I'm going to share my screen. Okay, so one of the reasons I came to this hackathon was to convert AlignTK to handle the 3D data that we have with the mouse data sets, the FMOST and the LightSheet. Given the limited amount of time, I only got about two-thirds of the way done there. So a couple more days, I think I'll have something that'll start to run. It'll probably need more tuning after that, though. But one thing I wanted to say was just the AlignTK was used for electron microscopy. So there's a lot of similarities here. The general strategy is to try to find a map that will transform the subject image into the reference image, or vice versa. And the representation for a map that I'm using is a grid type thing where you have XY coordinates on each of these, which tell where it goes in the target image. And in 3 dimensions, it will be XYZ going into a volume. And the way I'm doing this is using a stochastic hill climbing approach. I'm sorry if this is all messed up. I transferred my PowerPoint file from Linux to Windows, and it did all this strange stuff to it. But anyhow, there's a correlation term. I'm using Pearson correlation, which is comparing the subject image against the reference image. And what you want to do is try to increase the correlation and decrease the distortion. And I'm using a measure for distortion. So each individual grid element there is contributing to the distortion measure. And I'm just looking at the lengths of the edges and also the area of the grid cell. And I also have a term that can handle manually inserted correspondence points. Now, ideally, you wouldn't have to insert any manual points because that takes human time. But if you have a problematic pair of images, oftentimes it was useful to put in manual correspondence points, and this term let those have an influence on the overall energy. So this is the type of images that we were doing in electron microscopy in 2 dimensions, and you can see the alignment is fairly decent. One thing we did have to do was to eliminate a lot of these artifacts, such as these intensity artifacts, because this large image was formed by stitching together many small images. You'd end up with all these, very contrasty lines horizontally and vertically. And those cause problems to the registration. And that's why I kind of laughed when Min said that, they had to get rid of those too. So after doing an intensity correction, we would end up with something like that, which was much easier to align. And we did this in a multi-resolution way where you start off with one very coarse grid with one grid cell encompassing the entire image, and then refine that further and further to get down to not quite at the pixel level. Usually at the end, we'd have each grid cell representing maybe 64 by 64 pixels. And let's see. And it also worked fairly well on some nastier situations where you had sections that were torn up and we had to deal with those. So that I won't talk anymore about the 3-dimensional GPU-based alignment code, but as I was doing this, one thing that came to mind was one of the problems we're having at Bill is that we need to store things efficiently, and it would be nice if we could trim images down so that we weren't storing a bunch of noise. So in the FMOS data sets, this is one example. This is section 110 from the first FMOS data set in the set of subject data sets that Lydia gave us. And here's what it looks like just viewing it with GIMP. And you can see there's just a very small part of the overall area that's occupied by the brain. And for a section like halfway down, you can see this is about 5,000 something out of 10,000 something. So it's roughly in the middle of the data set. You can see the overall outline of the brain here. It's taking up a lot more area, but there's still quite a bit of black area along the edges. If you contrast and enhance these sections, you'll see something like this. And it shows that what's outside the brain is not just black, it is very dark gray. So it's not as though we can throw just black pixels away. We have to throw non-black pixels away too. So my question for the group is, how much can we trim off these images and set it to completely black and still have something that's going to be good to go into these registration pipelines? Now, this is what I trimmed from that 110 image. And the brain is this brighter region here. But as you see, if you set the exterior to black, you get a very sharp border here. The best way to handle this would be if you had a mask that also went into your registration pipeline. They would just completely ignore pixels out in this black region. And my Align TK can take these masks, but I don't know if other registration software can. And if you don't accept masks, then I would expect that this sharp edge here is going to cause some sort of problem. So that's sort of a question. And if you look at, here's a contrast enhanced view of the middle section. And you can see that the brain boundaries are here, and it's important to preserve these, I'm sure. You definitely want a few pixels of the blacks so that you have something that will align well to the reference image. But the question is, how many pixels do you have? How many pixels do you have to go out here to still have good input to your registration software? You probably don't need pixels way out here where it's completely far away from the brain, but you probably do need at least 5-10 pixels away from the edge of the brain. So that's what I'd like to find out. If you have any comments about how many pixels do you think you need, or how much of a distance relative to the overall size of the image, that would be useful for us to know. And also here you can see the effect of these. I don't know if they're acquiring these images in scan lines and they stitch them together or what, but these artifacts here are troublesome. They have to be removed. And if they could be removed earlier when they're doing the stitching, that would be a lot better than if we had to deal with them during the registration phase. So I don't know about the FMOS people, if anyone here can sort of speak to that, but that would be good to handle in software much earlier in the pipeline than during registration. Okay, that's all the comments I had. Did anyone have questions, or can anyone respond to this issue of how much do we really have to keep? Now, to give you an idea of how much we could save if we threw the exterior away, in this image right here, it was roughly 100 megabytes in size compressed, and the exterior took about 8 megabytes, and the brain region took about 92 megabytes. So we can't save all that much in this situation, but going up to the other one, this one, the overall size was around 65 megabytes, and the exterior was 50 megabytes. So that's a huge amount of storage that's really just going to waste. And the interior was about 15 megabytes. If we could only store the 15 megabytes, that would save us a lot of money. Okay, thank you.
Greg, I'd like to follow up on two of the things you said. How much black do we need outside the brain?
Mhm.
And how can we deal with trimming? So for the trimming issue, just a short question. I know the data set that Lydia shared was zipped. They were all .gz. So are you talking about data in memory or storage on disk? So
Storage on disk.
and so even though there is a .gz, it's still not doing a good job at, you know, compressing these large black regions.
Yeah,
Is that the case?
the compression of the exterior is not that great because it's not uniform.
Okay. And then, so in terms of how much space we need, would you mind if I share my screen for a second?
Sure, I'll stop sharing.
So just speaking for registration algorithms that use image contrast. So mBrain aligner identifies points, so that wouldn't apply. But if your brain looks like this, and you position your atlas over here as an initial guess, and we want to deform our atlas to match it, what's important is that this boundary lies in a region that we have data. So if we're only keeping this many pixels and this boundary doesn't lie in a region with data, then we don't know that we need to move that boundary. So that's sort of a rule of thumb. The amount of black we need outside is related to the amount of error that we expect positioning the atlas over this image.
Now, if that exterior was just set to black, would that cause a problem for you?
If it was set to black, it wouldn't cause a problem. But if it was set to, you know, ignore this data, this is outside the mask, that would cause a problem.
Mm-hmm.
Okay, I think only Alex is left.
I'm going to share my screen here briefly. I just had some final remarks that I wanted to make to everyone, and hopefully you can see. I just have this one PowerPoint slide here. I really want to thank everyone for your participation in the tutorial and for touching the bill system and doing some work on it, as well as all the folks who put forth a lot of effort into the tutorials themselves. I was able to listen to most of the tutorials and learn a lot from those tutorials that I didn't know already. So it's really interesting on that part. Just some general notes. We did receive a few questions about, will the accounts remain active? And yes, they will remain active. One thing to note though is that you will no longer have the reservation if you're submitting a job through Slurm or Interact. And really all you need to do is just don't use the -- reservation and the -a parameter, and you'll be okay with that. If you have any questions or issues with the bill system, want us to install some software, please feel free to continue to contact us at billsupport@psc.edu. One thing that we would like to be able to do is to make some of the codes that you guys have worked on and the workflows available through the Brain Image Library for other people to use. And we'll be following up with you on that, because I know from this presentation this afternoon that there's some tightening up that needs to be done for several of the codes. So we'll hold off on that, but when you're ready to make your codes available and want us to make them available to the outside world, we'd appreciate that that you reach out to us at Bill Support. We also have thoughts of incorporating some of the registration algorithms into a future data usage workshop, targeting, let's say, people like Iana who want to run a program but don't necessarily want to develop a registration code. And if you're interested in that or teaching in such a workshop, please contact me at Bill Support as well, and would be happy to discuss some future possibilities. Overall, it's been a great experience working with the participants, and a lot of the input that you've given us will help us prioritize some future enhancements at the Brain Image Library, such as making Jupyter Notebooks easily available through the Bill system. We already have Jupyter Notebooks are available through Bridges 2, and this has been on our list of things to make available at Bill. I think this hackathon really emphasized that it is something that we need to do and work on sooner rather than later. And finally, I would like to thank especially Lydia and Matt for their hard work, both in reaching out to the Bill team, as well as many others that are on this Zoom session right now to make the hackathon happen. I think it's been very successful, and look forward to the next one.
Thank you, Alex. Do you want to say your final words?
Yes. And so, yeah, I'd like to conclude with a few additional thank yous, and then a couple comments on what's next for us. So first, thank you to everyone who participated. It's been wonderful talking with you and working with you, and I am very impressed with everything that has been demonstrated and done together here, and I love to see the collaboration that has happened here. And I know Lydia is not here right now, but a special thanks to Lydia, who really helped organize this, and the folks at the Allen Brain Institute. A special thanks to Alex and Greg and Luke and Ivan and the team at Bill. I think it has been a great opportunity to have these compute resources, but moreover to have the guidance on how to use them and use them well. I've been very impressed there, and thanks for the help in organizing too. Special thanks to the BICCN and Lydia and Ricardo, who helped to provide our challenge data sets. These are excellent data sets here. Special thanks to Mark, who's in the room here at UNC, who helped provide an opportunity for us to come together in North Carolina. We will hopefully do more of that. I'll talk about that in a second here too. Additionally, the tutorials were very impressive and outstanding. So thank you, especially to Ivan and Jeff and Min and Lydia and Ricardo and Victor Vandervalt and Daniel and Gary and Ken and Jenan and Hastings and Caitlin and Bryson and Michael Miller, who provided these tutorials. And finally, yeah, thank you to everyone who provided your results and shared your lessons learned. Very impressive, and I think it was very interesting to see the commonalities and the differences, and kind of what worked well for folks. So what I would like to suggest for next steps is, I know many people made a lot of progress and they learned a lot, but they have a few follow-up work to do on the challenge exercise, which is expected and makes sense. And as you get your pipeline ready to share with the world, yeah, please do email Alex when you're ready. I will send an email out to everyone who registered and to the mailing list that was linked to on the website if you want to sign up for that in a week's time, and ask folks if there's more people who need more time or where you're at with your analysis, your registration pipeline. And then based on feedback there, we'll find a good deadline to help guide folks and see if we can have a summary paper at that point put together. And I would also encourage you to stay connected on the mailing list, and however you found out about this hackathon, so that we can do it again together. We were stymied a little bit for any in-person events by COVID, but the next event will be more in person. And I think the Gather Town and the Zulip was successful, and the Zoom was helpful for having a at least semi-virtual event. So we'll work on that too. So thank you again to everyone, and we'll see you again soon.
Thanks a lot, Matt.
Thanks, Matt.